Okay. So I'll not explain. I'll explain when we're talking. Okay. Now, so we were saying that there are some soluble factor in the sperm. People think they are present here, and they think those soluble factor can activate either PLC gamma or they can activate SERT kinase. So two views are here. So, but no one knows what that soluble factor is. But later what people found, they found when the sperm, so we are talking about the soluble factor in the sperm. So soluble factor, they believe that, so how the idea came about the soluble factor, that is an interesting thing. Think about the, we'll talk actually later about this thing, in vitro fertilization. But there are two ways to do in vitro fertilization. One way is to put the sperm and egg together. The other way is to inject the sperm directly inside the oocyte. So when sperm and egg are put together, okay, you can say binding is attached to EBR and this cascade start. But when you put sperm directly in the egg, then that is not going to happen, right? So still the development starts. So idea here is that there is something in the sperm that might be kind of, because G protein activation takes place by the receptor, right? So if the receptor is not there, G protein activation is not taking place, that means there is some sort of other thing which are activating there. So either the SERT kinase is directly activated or PLC gamma is directly activated. That is the view, right? But what they have found when sperm contacts egg jelly, so that contact itself, that increases in the sperm. What it increases is nicotinic acid adenine dinucleotide phosphate, and the concentration of this is increased by tenfold. So NADP, and this is called, in short form called NAADP. So when the sperm contact the egg jelly, the concentration of, in the sperm this is present, NAADP, its concentration increases tenfold. And what they have found, that actually NAADP, it play a critical role, NAADP play a critical role in calcium release in the muscles. So if they increase the level of NAADP, the muscle, so muscle contraction takes place because of the calcium release. So calcium does so many things. What they found that the muscle contraction takes place, and they found that calcium is released from the stores. So it means it can directly stimulate them. And the other thing it does is neurotransmitter release. If you remember, neurotransmitters are present in the vesicles, and those vesicles need to fuse to the synaptic terminal. So that also involves calcium, right? So what they found, that there are multiple scenarios in which NAADP, it can basically release the calcium from the storage. And then they thought that NAADP is probably the soluble factor which is present in the sperm, and that can cause the release of the calcium. Now, when the normal fertilization, and that's why they say like the cytoplasmic injection, intracytoplasmic injection of sperm is successful, right? But in the normal fertilization, that means you are having two waves of calcium increase. One wave is when the when the receptor is bound to the binding. That is going to stimulate the calcium. And then when the sperm is going to release its content inside the egg, that means you will have a lot of NADP that is going to further increase the calcium. That is making sense? So that's how there are two ways of calcium, two ways by which the calcium can be released. Now, what are the effects of the calcium release? So, effect of calcium release. What are the effects? So we can have two types of responses. Number one is called early response, and number two is late response. And the calcium level increase. So the calcium goes up within 10 seconds. Within 10 seconds, the calcium goes up. And when we say that calcium goes up within 10 seconds, so the calcium concentration increases from 0.05 micromolar to 1.5 micromolar. So, huge increase in calcium takes place there. Now, so when we talk about the effect of calcium release, the early response, that takes place within 15 to 60 seconds. So calcium increases in 10 seconds and all the early responses are taking place within 15 to 60 seconds. So the first fast response that happens is, number one is the cortical granule reaction. That's the first fast response. We know that happens, we know how it happens, right? And then what happens, the second response is, that is activation of NAD kinase. Activation of NAD kinase. Now, what it is going to do? NAD kinase is going to convert NAD plus, and here is NAD kinase. The enzyme is now activated, and when that enzyme is going to activate it, that is going to make NADP plus. Okay. Now, this NADP plus acts as coenzyme in lipid synthesis. So NADP plus acts as a coenzyme in lipid synthesis. Now, why we need lipid synthesis? Because we know after that the cell is going to divide, right? So if the cell is going to divide, therefore new membrane synthesis is going to go up. So need to make new membranes, because if you want to have, so because the cell is going to divide after this, one cell is going to give you millions of cells. So that's why you need a lot of NADP. So this thing is done by NAD kinase activation. Now, this NADP is also, that's the one way, that's the one pathway take place. Then the second pathway happen, that this NADP is actually changed into NAADP. And we know that NAADP also acts as a soluble factor, right? So it means, if here it is making NADP, that means you are having more calcium formation take place, more and more calcium is coming out. Okay. Now, that was the second round. Okay. The third thing can happen this NADP is utilized to make NADPH. Okay, now what is the purpose of this NADPH? Okay, so we said what happens when we have calcium release? We said that triggers CG reaction. And CG reaction, we said that you have UDX1 enzyme, right? And we said UDX1 enzyme convert oxygen to hydrogen peroxide, right? So think about, so oxygen is converted to hydrogen peroxide, and you have this UDX1 enzyme. So that means at that point, when oxygen is being converted to hydrogen peroxide, you need a lot of oxygen, right? So that means the calcium release, it increases oxygen consumption. Making sense? Now, here is what happened. So, I don't know, I think we covered in the cell biology, I don't know whether you guys covered it or not. Are anyone working with Dr. Reynolds here? No, okay. So what happens, I'm telling you because that should be the part of that what people do. So oxygen normally is reduced in the mitochondria, 97%, almost 97%. But almost 3% of oxygen, 3 to 5% of oxygen convert into this thing. It's oxygen with a lone electron there. And this is called superoxide radical. Okay? Now oxygen, this superoxide radical is really toxic because it's a free radical, it's an oxygen free radical. Free radicals can, if you remember your chemistry, are highly reactive species. They can react to anything, protein, lipid, DNA, RNA, so they are going to damage. So the cell has two defenses against this. One is in the mitochondria because they are made in mitochondria. In mitochondria, there is an enzyme in mitochondria. There is an enzyme called MNSOD. Okay. That MNSOD is going to convert this oxygen free radical, this is in the mitochondria, convert that into hydrogen peroxide. And SOD means, the full form of SOD is super oxide dismutase. Okay? Now, some of this will be changed into hydrogen peroxide in the mitochondria. Some of this will come out of the mitochondria. There also you need to detoxify it. In cytosol, this is Mn. Mn is manganese. It's from manganese. Manganese is a good factor. In cytosol, there is an enzyme called copper zinc SOD. It utilizes copper and zinc as the cofactor. But what it does, it also does the same thing. It converts oxygen to hydrogen peroxide. Okay? Now here, the UDX1 enzyme, which was in the cortical granule, is doing the same thing, right? But what is there, hydrogen peroxide is a highly toxic thing. It's a pro-oxygen. It's going to do all the oxidative reaction, going to kill actually everything in the cell. It's good for the purpose of kind of like, that's how it does it's solidifying in cross-link because it's making those bonds. But you don't want your proteins in the cell to cross-link like that. So for that purpose, the egg developing egg need to protect the cell from the hydrogen peroxide. right? Every cell has what you call is, I'll show here how we use it. It's called glutathione. It's okay to wipe it, right? So every cell has glutathione. Glutathione. It's called GSH. Okay. This glutathione is there. This glutathione is utilized to neutralize hydrogen peroxide. Okay. So at this stage, what happened? There is an enzyme called glutathione peroxidase. Glutathione peroxidase enzyme is there. What that glutathione peroxide, this enzyme, with the help of glutathione, convert hydrogen peroxide to water. Hydrogen peroxide is converted to water. And then it changes this glutathione to GSSG. This is oxidized glutathione. Okay. Now, this oxidized glutathione cannot be utilized. It cannot be utilized for neutralizing this. Okay. So, but you need to put it back to this form so that all the hydrogen peroxide which is produced there can be neutralized, right? You need to protect the developing egg from the oxidative damage. So for that purpose, there is an enzyme right here which is called glutathione reductase. Okay. That enzyme utilizes NADPH, and that NADPH will be utilized, that will convert to NADP in this process so that you can get the glutathione. Right? So that is the third thing that is happening. So we are saying NAD kinase is activated. NAD kinase make NADP. NADP is utilized as the cofactor for new membrane synthesis, right? And we are saying that NAD is also changes into NAADP. NADP is changed to NAADP to get more and more calcium. And then we are saying this NADP is changed into NADPH. And NADPH is utilized to maintain the concentration of glutathione so that cell can be, or the developing embryo can be protected against the oxidative damage. That is making sense? Or no? Okay, talk.
I'm having a hard time getting it. Will you repeat it one more time?
Yeah, so what we are saying that there are, first early response was cortical granule reaction, right? Second early response, we said the activation of NAD kinase. What NAD kinase was doing, it was changing NAD plus to NADP, right? And we are saying this NADP act as a cofactor in the lipid synthesis. Lipids are required for making new membranes. So this one cell, which is fertilized egg, this is going to divide like crazy to give all the cells of the body, right? It means you will need more and more membrane. That means you need more and more lipid. That means this NADP will be utilized. That's one of the first things. Now this NADP, which is made there, this NADP is also converted to NAADP. NAADP we know is acting as the releaser of calcium. So that means you will have more and more calcium being released there. And then we are saying this NADP is also converted to NADPH. And we are saying NADPH is required to maintain the concentration of glutathione in the cell, because it is going to help the enzyme glutathione reductase to convert this oxidized glutathione into reduced form or the normal form of glutathione. And we are saying why that is required. We are saying because when the calcium is released, burst of oxygen consumption takes place. Because you have then you have the UDX that is converting oxygen to hydrogen peroxide. Hydrogen peroxide is deleterious, damaging. So you need to get rid of that. To get rid of that, you will utilize glutathione. That means then you are going to reducing the amount of this because this is converted to this. But you want to maintain this. For that purpose, you need NADPH. So that's the other process. Making sense? So if we talk to each other for a second or a minute or so, then we'll go over.
He said that they both have it, right? Like, in sperm contact, releases like the membrane of it, and then like opens up or whatever, it also releases hormones. Right? No, but don't question. Because we were talking about these two. I thought they were like dudes, like, I don't know, like one or the other half. They both have them. But, because this is the membrane one, or like you just talked about, this is the membrane one, and the other one is the soluble factors.
So maybe we're just looking at the soluble factors.
I think they both have it. I think the membrane happens first, it's like the early response.
My understanding is that, yeah, these are the two views, but they each end with initiating calcium. And then after that, they both think the IP3 plays there. So it's just getting to,
Because when we talk about it, and we both, I mean, at least we discussed
but
it.
I think that we already discussed this one.
So that was
So I
like,
think it's just
or is that
different.
right?
Yeah, so this side thinks that whatever we just talked, like we talked about previously, thinks this is how calcium is released. And now we're talking about how the people that think the soluble factors.
Okay.
That's my understanding. So then the effects of calcium release. So with one early response, calcium increases within 10 seconds. The concentration increases from 0.05 micromolar to 5 micromolar in 15 seconds. And then it has the other one is late response. We can talk about it. And then
I thought that was, I thought this was the response.
So, is all of that stuff that we just talked about late response or early response?
We just talked about early.
Oh.
So far it was
So
all
we
early.
haven't done it late.
No, we have not touched the
Okay.
late. This was all early
Okay,
response.
just making sure.
Yeah, this was all early response. Remember, UDX1 is the part of the CG reaction.
Okay. Okay, so then the cortical granular reaction, which is what we talked about on Friday. So, and then 2 is the activation of the NAD kinase. So,
So these both happen, right? Never mind. I don't know what I was about to say. Never mind. Okay, never mind.
So then NAD plus is converted by the NAD kinase to NADP plus. And there's three things that this can do. So one, the first one is acts as a coenzyme in lipid synthesis and new membrane cells. Or those make new membranes. And then number two, change into NAADP, which is a releaser of calcium to increases the calcium. And the third is it can be converted into NADPA, which is important for this step of converting G and like DSH from its oxidated state back to its normal. So then we talked about high calcium levels plus the calcium reaction, which increases the O2 consumption. So then O2 is converted to hydrogen peroxide by UDX, and then but 3 to 5% of the O2 that isn't converted to hydrogen peroxide is converted to superoxide graphene. But I didn't understand.
And this is required enzyme.
I think it's just a little extra. That's what
I was like, so that superoxide is
so this NADPH is
bad.
required.
So the cells, the cells have two defenses against it. The first one is the mitochondria, so the
If this is
MUSOD.
not there, then it's too low. Then it makes sense. Right? So that's why we need NADPH. Makes sense?
So that is converted to hydrogen peroxide. Then we can
Yes.
use my
Yeah,
little
mitochondria has
What do you call
NADPH,
it?
superoxide disturbance,
oxide
Yes.
dismutase, but that's only for that. So it's magnesium superoxide
Yeah.
dismutase.
I went to write it and I was like, I don't know what that last
There
part
is a
is.
string.
Okay, and then the other one is the cytosol, the
So
copper
there should be
zinc
a dot here.
superoxide dismutase. Can
That's the
also
three
convert it.
electrons.
What does right? I don't see it right now.
So this is the other, this is the second one. Cytosol,
Anything else?
copper,
And they both convert.
yeah.
Any confusion here? You good?
Yeah.
You guys are good here?
Yeah.
Okay. Now we are going to talk about the late response. Okay. So late response, what happened? Remember we said when we have fusion of egg and sperm, that lead to via activation of DAG, not activation, so formation of DAG. Right, formation of DAG take place. Then you had the activation of PKC because it activates PKC. And then you have calcium go up. They both go to sodium proton anticoder, and ultimately that leads to increase alkalinity of cell, right? And we said what that does when that happens. You have number one, nuclear membrane breakdown, and you have DNA synthesis starts. Now all that is happening because calcium is also playing a role there, right? Now, calcium is actually, so if you take a cell and just increase the alkalinity, this is going to happen. But calcium play a critical role in starting a new DNA synthesis, okay? So calcium increase is critical for new DNA synthesis. that is critical. And what calcium does, it actually inactivates mitogen promoting mitogen activity promoting kinase 1 or MET kinase 1. Okay. Now, this is a very interesting kinase. In cancer cell, that's why every cell is different. If you talk about the cancer cell, you inactivate this kinase, you will stop the cell division. But in this case, where we are talking about the development, it's actually inactivity. Once it is inactivated, MAP kinase is inactive, what it does, it actually leads to DNA synthesis. So this is very different from the other cell types. So here, its inactivation is important to start the new DNA synthesis. In cancer cell, it reverses. Okay? So now, the other thing happened, that's the first thing. Second thing is protein synthesis starts. Now the protein synthesis starts from where? Protein synthesis starts from the existing mRNAs in the egg. Remember when we were talking about the egg, we said there are thousands of mRNA stored, right? And those mRNA are stored once the calcium level up. What that does, that starts the mRNA, to utilize those mRNA to make the new proteins. So it means for the initial protein synthesis, no new mRNA is synthesized. This is critical. So there is a thing called actinomycin. Actinomycin is inhibitor of transcription. So if you take actinomycin and you put it there, it's essentially going to tell you that whatever translation is taking place, that is taking place from the existing mRNA. Okay? So no new mRNA is synthesized. Just remember that no new mRNA is synthesized in the initial translation process. All the mRNA are utilized which are already present in the egg. Okay? Now, what kind of proteins are being synthesized? Right here. What do you think? Say it again.
You said it was an inhibitor of what again?
Forget about that.
Oh.
Don't confuse yourself. Just remember that. That's why I didn't draw. I was going to draw a graph here. I don't want to confuse you. Just remember that whatever protein synthesis takes place, that is due to the mRNA already present in the egg. So no new mRNA is synthesized during this process. Okay. So what is, because think about that. I'll explain why it is not synthesized. I have too many things to talk. So just think that no new mRNA synthesized, protein synthesis takes place. So what kind of proteins are made here? Histones, tubulin. Because what you need to do, you need to start the new cell division, right? So you have started making DNA, but now synthesize the DNA, but DNA doesn't exist as DNA in the eukaryotic cell, it acts as chromosome, right? And chromosome have DNA and histone together. If the cell has to divide, you have to make the mitotic spindle, so you need tubulin proteins, right? And then you also need what we call morphogenic factors. Morphogenic factors. Morphogenic factors are required for early differentiation. You want to make cell A, B, C, whatever they are going to be. So you want that also. So this is going to happen because of the calcium rise. Now, remember when we were talking about in the beginning, we said there are thousands of mRNA, but those mRNA are silenced. I mean, they were in the egg, right? But why they didn't do any protein synthesis at that time? So there was a reason, there was a block on that. That block is removed by the calcium. And how that happened? So what is going to happen there is, if you remember from your class in genetics, you remember that you have this mRNA there. mRNA is 5 prime, 3 prime just drawing like this, right? If you want a protein translation to start, you need an elongation factor. And if you remember, I think it doesn't exist like this, it exists like this. I think you can open your notes, previous notes, and we draw this in genetics, how the translation starts. So there are many factors right here. This is 5 prime, this is 3 prime. Here, at this site, you need to attach a translation. You need to attach a protein. And that protein is called translation initiation factor. A translation initiation factor should be there. And that is EIF4E. So this should come and bind here, right? But what happened in the egg? If it binds here directly, it's going to interact on this side. There are certain things, I'm not going in that detail. But purpose here is that it should be able to bind here. If it binds here, it should start the translation. But it cannot do because there is a EIF4E binding protein. That binding protein is kind of, if this is this, right here, right here. So this is not allowing this transcription initiation factor to interact with mRNA. Okay? So if it cannot interact with this, it cannot start the new protein translation. Making sense? Now what happens? Increased calcium, it actually activates cyclic AMP. And when cyclic AMP is activated, this cyclic AMP activates protein kinase A. Okay. Protein kinase A come here, and what it does, it phosphorylates. Is phosphorylate elongation factor for E binding protein. So what happened? Right here, it is a phosphate group is added here. So this is a phosphate group. It's phosphorylate here. Okay. So once it is phosphorylated, what happened? Phosphorylate F4, and that causes degradation of EIF4E binding protein. Okay. So now this protein, EIF4E binding protein, is degraded. It's not there. So now this elongation factor for E can interact with the 5' end of the mRNA and start the protein translation. Making sense? So we are saying that calcium bursts cause increase in the protein translation. That protein translation is independent of new mRNA synthesis. Whatever mRNA are present there, they make those tubulin, histones, and differentiation factor. But that can happen only if the block on these mRNA which were already present is removed. And what is that block? That block is a EI4E binding protein that protein binds to translation initiation factor EIF4E and block it to bind to the 5' end of the mRNA so that the translation can start. What happens when the calcium is increased? That activates cyclic AMP through adenyl cycle. I can go in that detail, more signaling. But that activates cyclic AMP. That cyclic AMP activates protein kinase A. That protein kinase A phosphorylated along with EIF4E binding protein. So once this protein is phosphorylated, this protein undergoes a degradation. Now EIF4E binding protein is no more there, so this translation initiation factor can interact with 5' end of mRNA and can stop the translation. Making sense?
So
Yes.
when the EIF4E binding protein is phosphorylated, then it starts being degraded.
Yes, then it is degraded. Yes.
So the translation initiating factor, is that also called EIF4E?
Yeah, that's the name of this. That is the name of this translation. That is the name of this.
Okay, and so the binding protein is that's associated with it?
Yeah, so binding protein associated with this. That's why we call it EIF4E binding protein.
Okay.
Once it binds, it doesn't allow this to bind. Okay. Now, so one of the protein which is, we talk about tubulin and all those things, the other protein which is made is cyclin. Remember, we know about the cyclins, right? So we said cyclin proteins are there, they regulate the cell cycle. So one of the protein that is made is cyclin B, right there. When cyclin B is made, cyclin B is made, and that cyclin B interacts with cyclin dependence kinase 1, CDK1, and once it interacts with this, that starts cell cycle. So that starts cell cycle. So if the cell cycle starts, that means cyclin B, this, and CDK1, when they are together, right? They together makes a mitosis promoting factor. So what we are saying? We are saying that once the inhibition of the translation is removed, new translation begins. Histones are made, tubulins are made, differentiation factors are made. Among those, cyclin B is also made. Cyclin B binds to cyclin-dependent kinase 1. Once it binds, it creates mitosis promoting factor, and that means it will start the cell cycle. That means the mitosis begins. So far it is okay? Okay, then what happens? So you guys can talk to each other, so make sure that there's no confusion.
The season of the egg sperm leads to the formation of that, and then the activation of PAC with increased calcium levels opens the sodium ion antiporter, which increases the alkalinity of the cell, and this results in nuclear membrane breakdown and DNA synthesis. So the histone, the tune, and the morphogenic factors are all things that happen in this thing. Like I'm confused.
Histones, tumor, and morphogenic factors in cyclin B are all made from the existing mRNA.
But then is that like this process within the
No, this process, so remember how like translucent shutdown? This process I think is like how it restarts the history.
So how EIF4E needs to attach at the 5' end to start the synthesis, to make like synthesis. But there is an EIF4E binding protein, which makes it so that can't happen. But then protein kinase A, which is activated by CNP,
Any doubt?
relates the cap, and that causes the cap to be degraded.
Any doubt here? No.
EIF4 starch absorption.
Any doubt here? Yes or no? Any doubt? No. Okay, good. So then, let's move forward. So we are going to talk about fusion of genetic material. So how that genetic material? We are again talking about CRG12, right? So what happened? That we are saying that so when the sperm and egg fuse, so flagella, mitochondria, they all sperm, they all regenerate. Okay. So now inside the egg, what we get a so this is, for example, the nucleus of the sperm. So the first thing should happen that the nuclear membrane should go over, right? And then the other thing should happen. So number one, nuclear membrane vesiculation occurs. That's the first thing. The nuclear membrane should vesiculate out. Okay, that's the first thing. And the second thing should happen, I'm talking to the sperm nuclei. The second thing should happen, remember we said the sperm chromatin is highly compact. And we said histones are replaced by what? What was there instead of histone in the sperm? Protamines, right? So very highly condensed. So what happened? That protamines should be replaced with histones. And remember, we said histones are already being made by the egg mRNA, right? It was making so many of those. Therefore, decondense chromatin. Okay. For the egg nuclei, you don't need to do this because they're already histones, right? But for sperm, you need to do this. But for egg nuclei also, you need to break down the membrane also, only then the next division can take place. Making sense? So now what is happening? So right here, if you remember, there is a matrix right here beneath the nuclear membrane. This is nuclear membrane, right? And this is nuclear lamin. Lamin proteins are there, which are intermediate protein there, we know this thing. And what happened, that when the calcium is going to get in, this is all done by calcium, right? We said calcium activates cyclic AMP, we just said cyclic AMP is there. We just talked previously, right? Cyclic AMP activate PKA, protein kinase A. And what that protein kinase A does, that protein kinase phosphorylate nuclear lamin. So this nuclear lamin will be phosphorylated. So this is all phosphorylated nuclear lamin. And you remember from your cell biology that once this lamin is phosphorylated, the lamin protein disintegrates. Okay? When the lamin is going to disintegrate, what is going to happen? That is going to cause the vesiculation of the, so this is going to lead the way, ultimately this nuclear membrane will break down like this, and you will have lamin will all remove from here. So this is lead to, or just right here, lead to vesiculation of nuclear membrane. Essentially, there is no nuclear membrane now. That forms small vesicles in the cells. Okay? So now what you have? You have here your DNA, and that DNA is wrapped around. So this is your DNA, and DNA is wrapped around proteins. So proteins are there. Now you need to replace proteins with the histone. Okay? Now what happened? Again, this PKA which has caused the lamin phosphorylation. Okay? So PKA cause phosphorylation of proteins. Now proteins are phosphorylated. So right here on these proteins, what you will have phosphate group attached. PO4 means 4 negative group, PO4 negative, PO4 on each of this. So it means one phosphate with this O, 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 and here it is bound with protamine. 3 negative charge on one protamine molecule. Okay? DNA is a negatively charged molecule. Negative charge on this, negative is going to repel each other. Okay? So that is going to lead to release of protamine. So protamines will go away. Protamine go away. Okay? And they will be, from here, they will be replaced by histones. That is making sense so far? So proteomines are now replaced by the histone. So decondensation, therefore, decondensation of sperm chromatin takes place. So we said activation of cyclic AMP take place. Cyclic AMP activation causes protein kinase A activation. Protein kinase A phosphorylate laminin. Once this laminin is phosphorylated, that causes vesiculation of nuclear envelope. Nuclear envelope is gone. Now this chromatin is exposed to the cytosol of the egg, and there pKa phosphorylate proteomine. One proteomine gets 3 negative charge from oxygen. DNA is negative charge, they are negative charge, so negative repel and proteomine go away. And then proteomine are replaced by histone, so that means the compactness of the sperm chromatin is gone. Now it's normal decondensed chromatin. Making sense so far? Now what happens?
We have 2 minutes.
Yeah, it will be quick. So what is happening there? Now we are saying that fusion of genetic material has taken place. Now, what is happening here? Right here is, so this is the, I should not draw the, I'm just drawing this round thing just for the sake of simplicity, but this is gone, right? Let me say this is just gone. There's nothing like that. This is the chromatin. This is sperm chromatin. This is now X chromatin. Okay. Now everything is happening kind of real time. You know, on one side, just think about, you had the membrane. On one side, this membrane is being removed. Side by side, this thing is happening. So what happened? Right here was centriole. Centriole come from sperm, right? So when the nuclear membrane is breaking down, the nucleus, the sperm nuclei, rotate at 180 degree. Okay? So when it is going to rotate, so remember we said the centriole also duplicate, right? Centriole also duplicates, so centriole is also duplicating, and this rotate like this. So this, the chromatin is kind of reaching here. So now one centriole will migrate on this side, one centriole is going to migrate on this side, and it's going to form spindle. And what happened? It starts new life. Because the cell division has started. So what happened? We are saying, as the nuclear membranes were disintegrating, histones were being replaced, right? So the sperm rotate, the nuclear rotated 180 degrees, so that they can move on both the sides. The centriole move on both sides, form spindles, and start the cell division. And that means the new life has started. Making sense? So talk to each other for a second or minute, then we will go to the lab now. So,